So it's a pleasure to have with us here today Ryan Kelly, who's an assistant professor up at the University of Washington. There's a number of reasons why it's a pleasure to have him here today. The first of which is that he is actually an alumnus of the ecology and evolution major at UCLA, so he is one of our own. After he finished at UCLA, he did 2 masters and a PhD at Columbia. And his thesis research was focused on population genetics, biogeography of marine invertebrates, particularly chitons. He started very early in DNA barcoding. And then he did a postdoc with Steve Palumbi at Stanford, where he did what I think is probably still the largest comparative phylogeography study that's ever been done. And after he finished his postdoc, he did what all people do. He went to law school. He's always had this strong interest in science and policy and the interface of that. And a lot of us scientists who dabble at this interface, we do our best, but he took it to the next level and really tried to understand the legal foundations of environmental policy. And he's been very active in writing about things like the Endangered Species Act and the use of that in environmental policy, as well as ocean acidification. And I forget you're in the college, what's?
UW?
Yeah.
College of the Environment.
College of the Environment. And
the School of Marine and Environmental Affairs,
okay.
Within the College of the Environment.
So he's still very much active at this interface, publishing both on the hard science side as well as the application of that science and policy. And he's one of the leading figures right now in the use of environmental DNA in marine ecosystems, which is what he's going to be talking to us about today. So, Ryan, thank you so much for coming.
Thanks, Paul. This is really a pleasure. I haven't been back to UCLA in a very long time. All of these buildings are new. This building is unrecognizable, and so this is very cool. And so, thank you. As I told Dave Jacobs before getting started here, I will always be grateful for being given the chance to start playing with DNA pretty early in my undergraduate career. And so that was 19 years ago, if we can believe it. So that is pretty cool. So thank you. And thanks, Paul, for that great introduction. I have a 2.5-year-old, and so I have to choose very carefully the number of hours that I can be away from home when I'm not doing childcare. And so these are those hours. So I've been using them well. I've had a great time already this morning. I think the law background explains the title a little bit here. Not just environmental DNA as a really cool, I think, frontier of how we learn about the world, and I'll talk in detail about that, but also what we do with that information from a policy perspective. Where does it plug in? Who has incentives to use that? And do they have the right tools to be able to do that? So that's sort of the both halves of my brain that I try. Often unsuccessfully, but I try and think with both halves of my brain when getting into these projects. So what may be really gross when you go for a swim, but what is really awesome when you are looking for information about the world, is that this is a soup of information that we're swimming in, and only very recently have we learned to use that information to good effect. Everything is constantly shedding DNA out into the water, constantly shedding cells through metabolic waste, but also damaged tissue and skin and cells and so forth. That's constantly degrading. And so what you have is, in fact, I think it was Emily this morning that described, she said the ocean, yeah, there's some water, but mostly it's just a matrix of bacteria. I've never thought about the ocean as, yeah, there's some water. But that is, but it's not just bacteria, it's also all the eukaryotes, the metazoans that are constantly shedding cells out in the environment. And we can pick up that DNA, those cells, sequence them, and use that information for ecology, for other basic science, but also for things like environmental impact reporting and thinking about human ecosystem interactions, hopefully. So I had a colleague, I'm mostly in a policy department at UW, and so I try and get science done, but then I get inquiries from my policy colleagues who say things like, you do omics, tell me about omics. And I thought, I don't know what that means. So I thought it would be useful to sort of situate this sort of work in the larger, for people who don't constantly think about DNA and RNA and protein, situate this work in that world. These are the relevant omics. Of course, you've got transcription, translation. You can look at the world through these different lenses. If you're doing that, if you're looking at DNA, you're talking about genomics. If you're looking at RNA, transcriptomics, and proteomics, excuse me. Perhaps obviously, I don't do those last two because I can't even pronounce the last one. I work on genomics, and so my question mainly is what genes are present. Now, you can do this for a couple of different purposes, and surely more than just these purposes, but I would separate this into, you could think about this from a diagnostic perspective. That's like the DNA barcoding. Which things are here? Maybe what things live in good versus bad habitats. Humans constantly have, silly humans. Humans have this idea, right, that there are habitats that are in better shape and worse shape. But then we have a very hard time explaining why we think that or measuring that in any real way. So if you were then able to go and ask in a pretty reasonably comprehensive way what's there, you might be able to more quantitatively put your finger on what makes good habitat versus bad habitat from a biological perspective, and things like, hey, where are most of the species, or where most of a species biomass? People who are looking for fish are especially interested in this because the ocean is large, sparsely populated, and hard to find things in, and so it's of particular interest to fisheries people who want to know where the fish are. You could also do functional genomics, which, as many of you probably know and have thought about, ask questions like, are the relevant genetic pathways in place to fulfill some function, like fixing carbon? And this is work, I work in the laboratory of Ginger Armbrust at UW, who does a lot of this work at large scales, asking precisely this question. How is carbon being fixed? How does that work in global cycles, global climate cycles? And then this very interesting idea that it's not really about the species complements that are present, but rather the functional complements that are present in a location. And so you can actually look at the world through that functional genomics lens and say, well, I don't care which species they are, as long as these metabolic pathways are in place in the ecosystem. It's a totally different way of looking at the world. So pretty cool. What I've been doing is entirely on this diagnostic side. So I should say, and it should be no surprise to any of you, that DNA as a sampling tool is not at all a new idea. All sorts of cool things have been done for the last 30 years as DNA sequencing, Sanger sequencing, and now more high throughput sequencing has become cheap and relatively easy. Cool stuff. These are colleagues at NIMS in Seattle that have done used genetic monitoring in a different sense for a long time. These are papers from the 90s and early 2000s. This is perhaps my favorite. When I showed up to do a PhD at the American Museum of Natural History in New York, there was a freezer full of caviar, and it was because they were barcoding the caviar, each one of which is a sturgeon egg, right? And so they were asking how mislabeled are the sturgeon eggs, and so it's just a rare lab that you show up and they have a freezer full of caviar. Things like shark finning as well, right? Which species do the sharks come from which the fins come, and then maybe you could tell something about the populations even, depending on what the underlying population structure of those species might be. So all very cool, doable with Sanger sequencing, one sequence at a time. And then in 2004, which microbial people have been working on this for a long time, right? Because if you're a microbial person, you know that only a tiny fraction of the microbial world is something that we can culture. So it's like this entire discipline of 100 years, of which I know very little, so excuse me, pardon me for oversimplifying, but everybody knew that you were only looking at a tiny fraction of the diversity that was there, but couldn't really do anything about it. And then it became relatively easy to sequence massively, and Craig Venter sailed around the world and published this in 2004, which is now sort of a long time ago in the DNA world and technological world. So microbial people have been doing this kind of thing for a relatively long time, but it's only pretty recently that we've found out, oh yeah, orcas shed DNA into the water too, and you can detect orcas in a liter of seawater. You can count things that aren't the whole things, but rather are the genetic remnants of species that have been there recently or that are much larger. And that's very powerful. So I want to distinguish between two techniques here. One, quantitative PCR, which I'd also include digital droplet PCR in this, if people are familiar with that. It's sort of a fancier, more expensive way of doing the same thing, which is very cool. Looking at DNA from the environment that got shed into the environment, doing it in a quantitative way, looking for one species at a time. So this is the needle in a haystack problem when you know what the needle is that you're looking for. It's usually invasive species, endangered species, things where it's important to be able to get the last of the endangered species and count it, or the first of the invasive species and count it. And I'll give examples of that. So that's one set of techniques, looking at one taxon at a time. The other, with sequencing, massive multi-parallel sequencing, is looking at hundreds or thousands of taxa at a time. The question there is, what's there? Not how many of this thing is there, but what are many of the species in the environment? Can you show me many of those all at once? That, however, is less quantitative because of the biases inherent in PCR. What ends up being the thing you got the most sequences from is not necessarily the thing that there is the most of in the world. So PCR bias distorts the counting of that, but nevertheless, the sequencing offers a really interesting cross-section of the world, from a liter of water or a scoop of soil, or actually you can do it from the air as well, looking at fungi, for example, in the air. What has made this sequencing approach possible is this breathtaking cost curve of DNA sequencing over the past 15 or so years. What cost about $8,000 is a log scale. $8,000 in 2001 is now a tiny fraction of a cent. And I haven't even updated this graph since 2016. So it's gotten cheaper. And this white line is Moore's law, which is often cited in Silicon Valley, right, as this sort of unapproachable standard of technological progress. It has become very nearly free to sequence DNA on a per base pair basis. That's remarkable. And what that means is we can afford to be a little bit cavalier in how we're sequencing DNA. I can sequence 1 million reads of DNA knowing that 900,000 of them are going to be identical. And that's okay, because I'm interested in the other 100,000. Here's what the workflow looks like for those of you who don't think about these things on a daily basis. It is a pretty boneheaded thing. It is exactly what you would do if you had to do this. You go out, you fill a jar with water. That is most of the work in the field. You then filter out all of the water and you keep all of the non-water on a filter. This is mostly cells. Now, what size filter you use does matter, but basically we're taking whole cells out of the environment. Fragments of cells, free DNA, all of that does exist, but we're letting that go because there's plenty of DNA in the whole cells that we're getting. We extract the DNA through your extraction method of choice. Those are all, you can use any one of them. And then you have to decide, am I doing the needle in a haystack qPCR where I know what I'm looking for, or am I going to do the sequencing, in which case you do a regular PCR reaction, massively sequence the fragment that you get. Spend a lot of time behind a computer crunching that, the bioinformatics, and then annotate to a known database we use BLAST, there are others. So that is how the workflow works. It looks so simple when you put it in a workflow like that. But the real trick here on the sequencing side, and actually on the qPCR side as well, is in the primer design. So as with so many other techniques in molecular biology, you design the primers and they're going to work not exactly as you think you want them to, but so it does take a fair bit of trial and error. But you can design, if most of the DNA in the world is bacterial and algal, and as I get into this, by the way, I have started protesting the use of the word algae because that's not a thing. Like, not only is it not a monophyletic group, it's like all types of completely unrelated things that for some reason we call algae, and I don't have a better word for it though. So it's still algae on the slide, but I'm using it under protest. But what you're interested in is animals, if you're interested in metazoa, then you need to design primers that not just amplify metazoa and that signal above all else, but also exclude all of the things you're not looking for. And so designing primers is a significant trick here. But if you do it right, it can give you really cool data. So I will give examples of these, but before I do that, sort of a policy question here. I think it's totally amazing that we can count fish with a liter of water, and then I can find orcas in a liter of water. That is amazing, and I hope I never wake up and stop thinking that that is amazing, because that is goddamn amazing. However, we already knew there were orcas in the world, right? That's not useful. It is amazing, but not useful. So then my question is this actually providing a benefit, or is this just a cool thing? And I think the benefits are a couple-fold here, particularly the qPCR approach is better developed, for at least from a utility standpoint. There you have vastly improved detection of rare targets. So if you are going to spend hundreds of thousands of dollars paying humans to go look for a fish, it might be cheaper to go take a handful of water samples, sequence those, and then if you really want to find the fish, send the human out to where you got positive results. That has been done, and that does work. Also, rapid quantification is a huge advantage of qPCR. You can do that fast, but the amplicon sequencing, the benefits were less obvious, but I think are now becoming clear to me. In ecology and in science in general, right, you always have this trade-off between depth and breadth. Do you want to sample at 10 locations in great depth, or do you want to sample at 100 locations in not great depth because you only have so much time and money? With this approach, I mean, I'm using water samples because that's what we've been doing, but it works for soil and air, whatever you want to do. You can multiply your number of samples without multiplying effort, and that's very powerful. You could do time series. You can do series in space and time. You could do those sorts of grids, things that you otherwise wouldn't have time and money to do. So I think that is potentially very powerful. So examples that I've not been involved in that you may have heard of, the Asian carp is by far the most high-profile example of this qPCR approach. These are two species of carp that are not native to the Great Lakes Basin. People very much do not want them to get into the Great Lakes proper, and so they have been monitoring very intensively the tributaries to the Great Lakes, notably around Chicago. And this group, headed by David Lodge, who is at Notre Dame now, at Cornell, and his lab, these are all alumni of his lab, have been prominent in publishing that, and also in working with USGS to get state and federal money to go use eDNA to survey effectively for those Asian carp, and that's worked pretty well. They've run into very interesting philosophical problems that you might not have anticipated, like when the sensitivity of eDNA is much greater than the sensitivity of humans in finding the fish. What happens when you get a positive result from the DNA, but you can't find the fish with a human? Is the DNA wrong? It's not clear, and so that's one of these open questions I want to add, I want to sort of leave you with, is that these are pretty different ways of looking at the world, and we're kicking the tires on these techniques, and everything has an error rate. But if the error rate of DNA, if the false positive rate is very, very low, the sensitivity is quite high, then people who are used to, which is all of us, who are used to being able to see and touch the things we study, are going to ask, well, where's the fish? Show me the fish if there's a fish there. But when the sensitivity of humans, when our ability to catch that fish is quite low, I think we routinely underestimate our false negative rate, right? And when you have a high false negative rate and a low false positive rate with DNA, you then run into this question of, well, what do you trust? And that actually went to court in the Seventh Circuit. And eDNA, to my knowledge, the one example where eDNA is admissible in court that has been litigated, and it did happen. So interesting, and I don't know, keep it in the back of your heads. This is the open question there. Also, the coolest name of any species, the hellbender, which I've never seen, but is apparently a big salamander that's endangered. It's in Appalachia. They've been using this sort of technology to count those, find them in various tributaries there. Salmonids. I moved to the Pacific Northwest, and my God, do these people love salmon. There is no social gathering that does not feature a salmon to eat. But also, in the rest of the country, the question is, like, are we having salmon or are we not? In the Pacific Northwest, it's what species of salmon are we having and what run from which river does it come from? I mean, you have to know a lot to go to dinner in the Pacific Northwest. And but many of these runs are endangered. They are not the ones, hopefully, that show up on your dinner plate. Many of the runs are endangered, and the NIMS, the National Marine Fisheries Service, spends an ungodly amount of money counting salmon. So if we can make that a little bit easier and cheaper, we will save a lot of money, which would be great. And maybe we can help some of these things recover. So we're working on that a little bit, which I'll show you. Others have been counting salmon as well. And there are many other examples of qPCR in relevant use. So just wanted to flag some of those, and also flag the fact that a lot of this is in collaboration with agencies. So when you're thinking about science, developing science that has some practical use immediately, the best way to do that is to work with the agency that's also your target audience. Rather than like going and working on something really cool in the lab over here and then going to USGS and saying, oh, hey, I made this really cool thing, you should use it. It does make a lot more sense to work with USGS so that they have bought into it, they've developed it with you, and then they're already using it. Like, you've already won in terms of science policy interface there. So those are qPCR examples. The amplicon sequencing examples are in the earlier stages. And this is work that I have been involved in to a significant extent. Not the Evans et al. Paper, but we went to the Monterey Bay Aquarium. This was our first go at this, and I think we would do this differently if we knew then what we know now. We talked a lot, I talked with Rachel and Emily about reinventing wheels, and with Zach, we did a lot of reinventing wheels, right, of people, problems that microbial people had long since solved, that we were sort of coding by hand ourselves. But went to the aquarium. It's a 4 million liter tank. We took 1 liter out of it and said, can we see all the fish in the tank? And the answer was basically yes. We could see all the bony fish in the tank. Our primers worked really well for those. So that was cool. And the fish that had larger biomasses, also we got larger numbers of reads out. So that was helpful because it made us think that we weren't doing something totally crazy. We then went out into the field, which, this is a controlled environment. It's a big tank, but it's not that big a tank. The ocean is a much bigger tank, bacteria notwithstanding. And so we swum transects, so had divers swim transects, and then took water bottles alongside them and asked, well, what do we see and what do they see? We were looking for vertebrates in particular. We saw all sorts of vertebrates, from cormorants and geese to surf perch and sculpins. This is rockfish. We're annotating these things to, in this case, the genus level. Often we don't get annotation. We get annotations to about the family or genus level, depending on the fragment of DNA. But in the blue here is the DNA, and this is in space, distance in shore in meters. And then the orange is what the divers were seeing. A couple important points here. One, the patterns are very close to one another in terms of their space. The different shades here, which is a little bit hard to make out, the different shades are actually totally different habitats. So this is seagrass, this is kelp forest, this is rock reef, deeper rock reef, and then out into open water. These are habitats that are within tens of meters, so hundreds of meters of one another. They're right next to each other in a surging intertidal environment, and this DNA is going nowhere. we have the spatial resolution here is something less than 60 meters. The habitats that are 60 meters apart had totally different vertebrate faunas, which the divers found and we found. That was unambiguous in the DNA. And so it seems like this is not only is the technology working to detect these species, but it's working at quite small spatial scales. The mechanism for which I don't know. Best suggestion that I've heard is that here you have whole cells that are being shed from these species that are very rapidly being eaten by microbes. And if that's happening on the order of minutes to hours, then that would explain this sort of non-transport of the DNA. It's not that it can't be transported, it's that it doesn't survive that long. Perhaps open to a lot of discussion about that, surely, if anybody wants to talk about it. So that's where we've been. We wanted to take this idea, try it out in a really controlled environment at the aquarium, then try it out in a less controlled environment in the field, and it seemed to do what we wanted it to do. And now I want to talk for maybe 10 minutes here about a couple of ongoing examples that I'm involved with a bunch of collaborators. The first, qPCR, this is salmonids and other fish in Skagit Bay in Washington, also in Hansen Creek, Alaska. And that's actually the example I'm going to give in the next slide there. Mike Tillotson's a PhD student in Tom Quinn's lab. Tom Quinn is like the person you go to if you want to know about salmon. He literally wrote the book about salmon. And then on the amplicon sequencing side, I'm going to give you this top example, urbanization's effect on nearshore communities. I made reference to thinking about, can we measure human ecosystem interactions using environmental DNA? And this is my first stab at doing that. Came out in about September, October, and I'm going to talk about that with you. But we've got a bunch of others. Assessment of the Elwha River recovery. This is the river where they took down a couple of dams, and they're spending a bunch of time and money looking at what a river recovery might look like. And we've got an eDNA piece of that. Effects of oyster aquaculture, which also the Pacific Northwest loves. And then my project I'm quite excited about is starting on Monday when my new postdoc shows up, looking at ocean acidification's effects in the field along a gradient of chemistry in the water, looking at the community level effects of OA and hypoxia using eDNA. So just a brief slide on this qPCR project. These data are about 5 days old, and so we're just getting them, and that's very exciting. This comes from Hansen Creek, which is in Bristol Bay. Lake Licknigick has this creek. Let's see if this video works. Yes, it does. So it's not much of a creek, let's be fair. It's not huge, but it has a lot of salmon. And why we are using this creek, how do we go back there, is that UW has had a field station there for many years. They count all of these fish. Oh, it's still going. Hold. There we go. They count all these fish, and they count them daily. So they know how many fish show up. They know when they spawn. They know how many die before they get to the top. They know how many leave. They know everything about this creek and these salmon. So if you want to quantify, you want to like cross-check your qPCR results, this is the way to do it, because we know how many fish are in the creek. So Mike Tillotson and another went up there, took a bunch of water samples. The yellow is, so these are the sites. The scale is tiny. This is, I can't quite read that. Is that 2. I don't know. This is about, the blue line is about 1 kilometer. So it goes into 2 different streams here up at the top. So we've got a bunch of questions that we wanted to ask. One, at these different sites, does the DNA reflect the number of fish above these sites in the creek? 2, is Site B the sum of sites C and D? Is this an additive phenomenon where downstream you're just going to add all the DNA from above stream, and that you're going to see that? What is the degradation like over this 1.5, 2 kilometer reach? Or if there's not a lot of degradation, is Site A just the sum of all the DNA above it? And we're just starting to get data in. This is a representative slide. All the sites actually look like this, where yellow is the salmon run and gray is the qPCR results for those salmon. It's a very strong relationship, but it's shifted in time. So that's interesting, because I don't know why that might be. It might be that most of the DNA is coming from spawning. that most of the DNA is coming from gametes. It takes a few days to get up to the spawning ground and then spawn. We'll see if that happens. To a first approximation, we haven't done the math yet, but it looks very much like site B is additive between C and D. Those are very close to one another, but site A is lower in such a way that the DNA is degrading over that 1 to 2 kilometers, which is what you'd expect from the marine examples. Stuff is degrading quickly. So the picture we're getting here is that eDNA is giving you a pretty here and now view of the world and not integrating very well over time. So much more to come on that, because we have a lot of interesting cofactors like how much UV light is coming in, what's the temperature of this, how many days of sunshine are there, and so forth. We've got all that data for this creek, so we're going to be building a model to try and get pretty quantitative about predicting the fish from the qPCR data. So then on the sequencing side of things, this came out in PeerJ in the fall. Shout out for PeerJ also, if you guys are looking for outlets to publish. This is an open access journal that is much cheaper than PLOS One, which I also enjoy, but has this a great, very rapid turnaround time. And yeah, just a really good experience if you're looking for a journal. This was a big project that was a collaboration with NIMS. They had an ongoing project looking at the effect of upland urbanization on nearshore habitats using a tow net, where they're counting all the invertebrates using thousands of intern slave hours. And we thought, well, we can go do this and grab a bottle of water and we can do the same thing very quickly, which of course, it took a while. But they'd set it up in a clever way. So this is a map of Puget Sound. You've got impervious surface cover here is in brown. So what this tells you is that near Seattle, and on the east side here of the Sound is largely heavily urbanized. On the west side, a lot less. And so they have a paired study design. These are watersheds. So from the whole watershed, they took pairs of watersheds that were about the same size and about the same latitude to control for gradients in latitude that we knew happened anyway in Puget Sound. One that's heavily urbanized and one that's not. And so paired sampling design, what ended up in the paper was 4 pairs of sites, eight sites total, varying from 1% impervious surface cover to 42%, which is a lot of concrete. So these are all nearshore eelgrass habitats, Zostra, Marina, soft sediment, and we took 3 water samples per site, which they were taking transects, we were taking a bottle of water. So that's our level of replication. There we have, at the transect level, we did 4 PCR replicates per sample. So we actually have 12 PCR 12 things to sequence per site, geographic site. And we did this with DNA primers, mitochondrial 16S. I designed these to amplify metazoans, but exclude everything else, non-metazoans. That is key because most of what you get if you use a lot of these primer sets is diatoms, and there are a lot of diatoms in the world. So we sequenced it to about 200,000 reads per replicate. So with a tiny fragment of DNA, 116 base pairs, but it allows us to get a bunch of phyla. I think we ended up with something like 11 or 12 phyla that we ended up seeing. And here's what we found, much to our surprise, that as watershed imperviousness increased, the diversity of DNA that we got was increasing very strongly. We expected the opposite, right? We expected everything goes to hell, but no. Humans in this case are associated with greatly increased diversity. That's true if you're measuring OTUs, which is this solid red line. It's true if you're measuring taxonomic families, which is this dashed red line. It happened in 4 out of 4 pairs of sites, so that we were pretty confident in this result. I tried to destroy this trend in a lot of ways, and I couldn't do it. You can take the top 100 most common OTUs. You can take the bottom 500 most common OTUs. Same trend. So this seems to be real. Because you are all biologists, you will want to know, well, what are these magical things that we're sequencing? They are mostly arthropods and fish and mollusks, but with a bunch of other things thrown in, a bunch of bryozoans, echinoderms, and so forth. But a pretty cool first effort, I think, in looking at human ecosystem interaction, trying to quantify that, and saying, how does urbanization affect these nearshore watersheds? And of course, the headline in the newspaper, I think it wasn't literally in the newspaper. It was on the radio, though. There was a headline, and it was like, humans help ecosystems. And I thought, I'm not sure that's what you want your headline to read. And because, so the alpha diversity is increasing, but the beta diversity, is the next slide I'll show you, is decreasing. This is just showing you that it's almost entirely bivalves that is driving this. This is soft sediment habitat that is, humans appear to be greatly subsidizing different kinds of bivalves that are living near human settlements. And that's quite interesting. That's driving the vast majority of the trend. Beta diversity is a complicated thing that means many things to many people. So I want to be very clear what I mean by beta diversity here. This is within a site between transects is what I'm talking about. How patchy are these environments? So as you swim from one transect to another transect, how different is that sample likely to be? And in the less urban environments, they're likely to be quite different. So they are less rich, but they are more patchy. And the human-dominated ecosystems, the more urban environments, are richer but more homogeneous, less patchy. I think about this like I've been talking about as the mall effect, the difference between a small town downtown where you don't have that many options, but all those options are different, and a mall where you have lots of options, but they're all kind of the same. That's what we're looking at in Puget Sound, or at least with respect to the species that we picked up with these particular primers. Now, how did it compare to the people that we were working alongside who were counting things manually? Totally different pattern. They have no trend whatsoever. And so we have philosophical problem number 2. What happens when the DNA tells you one thing and your eyes tell you another thing, or what's in the net tells you a different thing? And this leads us to, and this is a comparison. The green is what was in the net, the manual counts, and the yellow is what we saw in the DNA. They're very, very selective, right? What shows up in the net is only, and they bother to count, is only things that are bigger than about this and pretty much fall into one of 3 bins. It's a handful of fish, and then mostly arthropods and mollusks. It's mostly shrimp, and a handful of snails. So if you look at those taxa, you get no trend. If you look at those same taxa in the DNA, you also get no trend. But we're looking at totally different taxa. None of the bivalves that we're looking at show up in the net. That's what is really increasing when you look at the urban signal, right? And so we're looking at different suites of biodiversity, and we get different answers. And that was very unsettling. But now, having thought about this for a year, I feel okay about that because we're using totally different techniques. We are looking at totally different swaths of biodiversity, and it's okay that those have different trends. It just was unsettling at first. Our primers, for an example of this, and I think it sort of says a lot about eDNA as this is, developing as a technique, is I gave a version of this talk earlier on, maybe a year ago, to Ginger Armbrust's lab, biological oceanography lab at UW. And I said, well, our primers, we get 11 or 12 phyla, but we don't get any annelids. They don't amplify, we don't get any worms. So I give the whole talk, and she asked a question, the PI asked a question at the end. She goes, yeah, but you didn't get any annelids, so how do you didn't sample biodiversity? And I said, well, but the people collecting in the net, they also didn't get any annelids, and nobody asked them that question, right? We've always thought that the things that we go out and count in the world is a representative sample of what's there, and of course that's untrue, and that's never been true, but we've just ignored that. They are wrong, we are wrong, we're just wrong in different ways. And so how do we be wrong in a better way? I think the way to be wrong in a better way is to be wrong multiple times. And you can use those, you can use that information in an additive way. So we did this, a subset of these sites with 3 different genes, 16S, CO1, and 18S, compared it to the manual counts. You will notice the 18S and the manual counts have 0 taxonomic overlap. These are taxonomic families. The reason for this is that 18S is getting mostly diatoms and other, what I guess are known as algae. And the manual counts are counting mostly shrimp and snails, right? So yes, if we had sequenced this much more deeply, we would have eventually gotten a bunch of the taxa from the manual counts. But the point is that the biases, every sampling technique in the world is biased. But we often ignore that because we only use one sampling technique. But then when you do a bunch, you realize that they're all biased. They're all biased in different ways, and you have to figure out how to fit those together in your head. So that was a real lesson. This just came out in Frontiers in Marine Science, which is a more expensive open access journal, but also a pleasant experience. The overlap is remarkably small, considering that we know that the DNA is there. And this is all due to primer bias. So if you want to sequence everything in the world, good luck to you, because we didn't get close. Even with these 4 different methods of sampling the world, these sampling curves, this is 300, 400 taxonomic families within one geographic region in Puget Sound, and we didn't come close to having those lines flatten out. So if you wanted to sample the world comprehensively with DNA or any other technique, you're going to have to work a hell of a lot harder than we did. That said, 400 taxonomic families is pretty awesome if you're talking about sampling the world. And so we can do that, and you can do that repeatedly. And you could ask, hey, are human settlements impacting that? And in what ways? What are the taxa that are winners and losers? So I have a lot of optimism about how these techniques are going to pan out in the future. But I think we need to be quite aware that every technique has a limitation. It has lots of limitations. Every technique has biases, and you got to know what your error rate is. You got to know how it's biased, and in order to do that, we have to do it. There's no way of doing that in silico. Yes, you can predict in silico what your primers will amplify, but no, it turns out that doesn't actually predict what your primers will amplify. I think a lot of good lessons. We've learned a ton in the last few years doing this, and so I'm happy to talk about it with whomever. So for policy application as or any application really, by way of summary, with qPCR you've got really efficient detection and quantitative, but of course some of these details matter. The degradation, are you sampling in a flowing river versus a lake versus sediment, whatever, like resonance times are going to change. All of that is going to matter. Detection is probably going to be possible for much longer in time with qPCR because it's a much more sensitive assay than DNA sequencing, where you have the signal-noise ratio problem. Mostly what you're sequencing is whatever happened most recently, like the cells that just got shed versus the cells that got shed a day ago. There you've got this real benefit of easing depth for breadth trade-off, but that you're going to get different results from different fragments, different DNA fragments. You're going to have to know what to do with that. And then you have to be aware that as much as you want to be surveying all of biodiversity, you are not surveying any real fraction of biodiversity, probably. Whatever that term might mean to you. And then more specifically to the policy potential, I think that we've already seen some application of the Endangered Species Act with listed salmon species, for example, but also things like water quality. We actually have no idea as a society how well our water quality criteria under the Clean Water Act protect ecosystems. We have no idea what water quality criteria do for ecosystems. We think that they protect human health, and that's how the standards are set, which is great and valuable, and I wouldn't want to change that despite whatever might happen starting next week and to the indefinite future. But we have no idea how ecosystems are affected, and that's sort of remarkable given that we've had a Clean Water Act for 40-something years. In terms of Puget Sound, they spend a lot of time and money looking at status and trends of the sound. Is this a way that we could do that in a way that's more cost effective and therefore free up resources for other relevant work? Same thing here, surely, with Santa Monica Bay. Food webs, I think, is a great unexplored frontier of this sort of work. If you know that some species are increasing in biomass and other species are decreasing in biomass, and you have some a priori hypothesis about how those things are linked trophically, what's to stop us from doing a food web that's animated in time, a changing food web using just water sampling over time? I think that would be awesome. And then sort of where the lawyer scientist pipe dream overlap is everything major federal projects have to do environmental impact reporting under NEPA, but literally nobody knows how to do environmental impact reporting very well, despite the fact that an environmental impact costs like $1 million and takes years, and there are consulting firms that do this. They're measuring handfuls of species. How they are picked, it's not totally clear. And the measuring, like, how do we know those are indicating what, right? This would be a way, rather than having to pick a priori a handful of indicator species, you can just ask, well, what was the impact? That would be cool. So I'll stop there. I want to say thank you very much to all of these people without whom we could not have done any of this. Jamie O'Donnell is my postdoc who also designed our bioinformatics pipeline that is freely available on GitHub. Jimmy Carl is my master's student. Natalie Lowell is my former master's student, and all the rest of these are close collaborators. So that I'll take questions, and thanks for having me.
Tried to compare the biodiversity, say, of metazoans in sediment and in water column in the same place? I
haven't. There are a couple papers out there that have looked at total DNA in sediment versus in the water column. Those also came out of Notre Dame, I think. They're very attached to carp as a model species. And they found that the DNA is more concentrated in the sediment, but harder to recover. So I think there's a trade-off there.
I just think the sediment might give you a longer-term record.
Yeah, I think that's probably right.
So the two kinds of information will complement each other.
Yeah, absolutely. Yeah, Rachel.
How can we advance mathematical theory to get at like decay rates for DNA and stuff? Like, what are the collaborations that need to happen?
There are. What are the collaborations? I don't know where the collaborations are. There are a couple of papers out there that have looked at decay rate in different contexts, also from the Lodge Lab. The half-life in fresh water seems to be 24 to 48 hours that they've looked at. Philip Thompson did something similar in aquaria, a similar result. Lawrence Asubra, her paper out of Stanford from Ali Baim's lab, similar result. So we've had that's at least 3 data points. There might be one more that I can think of that have agreed that they've agreed on a pretty narrow window of time that the DNA, at least in whole cells, is remaining available. So I'm less worried about the decay because I feel like I know how it works. But in terms of math, sure. If you'd want to model that. I don't know who would be the best person to do that. I think in practice, though, degradation is wrapped up with transport. And because if you think of everything as a point source of DNA that's like sort of constantly shedding DNA in a sphere, and then all other things are similarly shedding DNA in a sphere, then as you move from one place to another, it doesn't actually matter what the decay was from over here because you've moved into a new place where there's new DNA that's dominating the signal. So if you think of it as a signal-to-noise ratio problem where there's some decay and some transport, it becomes a more complicated problem, and I think that's how the world is working. And that's why we apparently have no transport of DNA. It's not that the DNA is not moving, really. It's probably that it's getting eaten, it's degrading, but really what we're doing is moving to a place where there's a different signal that's dominating.
What about temperature? Like, wouldn't that affect metabolic rates of microbes?
Sure. And of the macrobes, right? Like temperature you should shed more when it's hotter. You should chew through carbon faster. All of these things, you can go down all of these rabbit holes. Like what happens if you're reproducing, and like if they're gametes, like what happens if a shark takes a bite out of you? There are all of these things, and I think my answer is that if you sample enough, that comes out in the wash. So that's why replication is important in space. I think that's why replication in time is surely important, right? You're going to see seasonal signals, but you also see when, like, there was a random shark bite. Well, okay, then you have a signal from damaged tissue that spikes, but you never see it again. You can distinguish rare events from baseline. I think that's my best answer. Yeah, Paul, and then back there, then Zach.
With these samples that were being taken throughout Puget Sound,
Yeah, great question. And because we thought we were sampling the whole watershed, right? Because we looked at the watershed and we're sampling sort of at the mouth of the watershed. It was almost entirely marine and near shore intertidal. So we got like barnacles and mussels, high intertidal species. We got a handful of things that are associated with humans. You know, like you probably have seen this result where you get cow. Unavoidably, some of that is from the reagents, the lab reagents, I think. But yeah, it's not a terrestrial, it's not a strong terrestrial signal.
That's even, did you sample at the surface where the fresh
It's like sort of just subsurface. Yeah. Yeah, it's back there, yeah.
I had a question about sensitivity. So you said before that sensitivity is pretty good, which sounds amazing. But I wonder if that is still sort of ecosystem dependent. Like it seems to me that it would be easier to find DNA in say a stream than 100 meters offshore in the ocean where things are blue and they're salt.
Yeah, I'm forgetting the equation, and someone in the room will know it. What is it? Sensitivity is true positives over false positives, or someone going to correct me on that? Yeah, let's call it, unless somebody corrects me, let's call it that. It's either true positives over false positives or true positives over false positives plus true positives. I think that's what it is. And that quantity can be very high independent of the environment. So in the example you give, in the open ocean, cells will be fewer and further between, but your ability to detect them is unchanged, right? Like if the question is, if you run into a cell, will you see it? And with qPCR, the answer seems to be yes. But you're right, you should get a much lower signal in the open ocean because it is a lower signal.
Yeah, I guess I'm thinking more like if you're trying to find a rare species or something
Yeah.
in the open ocean, the probability of sucking a cell up into your file might be lower than in the stream.
For sure. Yeah. So your likelihood of detection is lower, but your sensitivity is not different. Yeah, Zach.
Yeah, so it looks like we sort of, with eDNA, can solve the breadth versus depth, but there's this
Solved. Check.
repetition, and so if you wanted to compare two areas, and we only have a day, and you wanted to do an environmental impacts assessment, maybe you want to compare a marine protected area to a non-marine protected area, and you only can take so many samples because a PhD is so long. Like how do you solve that problem where maybe you have to sample every day? And
A PhD can be infinitely long. I think you've introduced a false constraint. How do you solve? Yeah. So you get back to the classic ecological question of how many samples is enough,
right.
Or what's the minimum set of information you want on which, with which to answer your question, and obviously question dependent. But one thing that we found is that it is really important to have biological replicates in the field because you'd like to be able to characterize where your variability is coming from. You're going to have variability due to PCR. You're going to have variability due to different bottles of water, even when they were right next to each other. You have variability due to season, due to whatever, day of the week. And some of those are variables that you want to worry about because you think that variance is introducing either bias or is going to be somehow misleading. And some of that is variability that you don't care about, like day of the week. We can probably ignore. So I think, in the example you give of like inside and outside an MPA, you probably want to ask, what am I measuring? What is it that I'm estimating? What's the size of the box that I'm drawing? And I'm going to measure variability within that box. So for example, in that Skagit Bay project that I briefly mentioned, NIMS is interested in counting salmon, but also forage fish and other. NIMS is a whole agency dedicated to counting fish, basically. And so their question is, can you help us count fish? And we said, maybe. And so we realized that if you take 3 bottles of water next to one another, you're going to assess the variance due to bottle of water in a very small space. But if you decide you're going to draw your box mentally bigger, we might decide the box is the whole bay or the whole MPA. So instead of taking replicates immediately next to each other, spread your replicates out and take 10 replicates over the whole MPA. You won't know what your variance is on a meter to meter basis, but you'll be able to say what's the variance within, between treatments, so inside and outside the MPA, and then you imagine your box plot is much more powerful, right, in that case.
Well, what about, if you're getting 60 meters, like what if you just sampled the awesome biodiverse kelp forests, like that rock just happened to have way higher biodiversity than the other one.
Yeah.
And it's changing on such a small scale, can we solve that problem?
But I don't think that's a problem. I think that's true. That's how the world is. So I hear your question, right? And it's hard because what you want is your error bars to be very small. Like you really want your error bars to be small. But the world has big error bars. So if you spread out your samples, yes, you will get the rock that has a lot of biodiversity, but you will get the rock that has like a little biodiversity. And the question isn't how small can I make my error bars? My question is, is my treatment different from my control? Right. So they will both have big error bars, but you will be adequately addressing your question. Yeah, Mark and then Deep. Yeah. All of the questions that we have geographically biased questioning. I haven't seen any hands over there.
A commenting question. So just on the aquatic toxicity issue, you were talking about regulatory and the Clean Water Act. The National Toxics Rule, half of it is aquatic toxicity.
Toxicity, yeah. So if it doesn't kill you, or it doesn't kill the fish.
No.
Yeah.
Specific toxicological outcome. But so one of the things that you brought up in sort of looking at the degree of urbanization impact on biodiversity reminded me of some work that was being done on the impacts of sewage treatment alcohols on biodiversity, biomass, and those sorts of things. Mm-hmm. And that same sort of effect was brought up on the board, and we both did this together many years ago, where sewage is good for the fish, but you
right.
looked at it in much greater detail, and of course, there's pollution-tolerant species that end up being attracted to those areas. So you get a combination of what you would expect in a healthy ecosystem with what you would expect in an unhealthy ecosystem. You add it together, and you get increased biodiversity.
Right. Yeah.
You guys looked at pollution-tolerant species, and seeing whether or not that's sort of where some of that came from, and looking at those bivalves, where a big group of them pollution tolerant, and then I'm also curious what you're doing on OA.
We don't, so the answer to the first question is no, I don't have a lot of that information. I didn't at hand, but I would be interested to know. They were all native bivalves. Like they're all bivalves that already occurred in Puget Sound anyway. So it's not anything that's like very different, but yeah, let me follow up with you on that.
Polychaete worms just went crazy.
Yeah, exactly.
Their conograms crashed. It was that kind of thing.
Yeah, there weren't any particular taxa that totally fell off associated with urbanization. So I looked at a taxon by taxon basis, and I would have expected that effect and didn't see a negative effect on any particular taxon. So that was as far as I got with that analysis. With respect to OA, the plan is Hood Canal is a gradient of dissolved oxygen, pH, and everything else. The outer coast has changes in pH and DO and other parameters driven by upwelling, seasonal upwelling. Inside a Hood Canal is largely driven by a flashiness of the Fraser River. So we've got different sources of sort of geophysical things going on that are driving differences in chemistry. And in the absence of a controlled environment where we can play with one parameter and not others, and keep others constant, we're hoping that we can sort of triangulate and say, okay, well, if these chemical parameters are all, if omega is changing, but it's changing because of three different drivers, but we see the same biological effects in richness, for example, that maybe we can start to attribute that to the change in omega itself. It's the best I can do so far. We're going to, as I say, we're starting next week, so we haven't gotten into the weeds yet. But it'll be seasonal, it'll be repeated time sampling over time and space along a gradient.
So on the last line you have in regulatory, the phrase you used was cumulative impact.
Yeah.
And that, as I'm sure you know, is a term of art in these EIR types of analyses. And pretty much everyone I've ever read was this sort of vague hand waving of, yeah, our project, yeah, other projects, yeah, not much to see here, move along.
Right, yeah.
Do you have a sense that these kinds of more broad approaches to looking at the first could give better answers, or answers that we could hold these analyses to give us a better sense of the actual cumulative impacts of multiple projects that today we don't really do very well with.
Yeah, it's a great question, and I don't underestimate the difficulty of that, because I think even given infinite time and resources and brain power, it's not obvious how to address the cumulative impacts problem, especially in California, right? Because under CEQA, you actually they're serious about it's in there, but how do you do it? Yeah, I guess my thought there was that. If you can document what species have been present in, say, reference ecosystem, reference sites, and then you can document the loss of those species or a change in that community as a result of like a series of development projects, then you could get at it at least in a better way than we're doing it now. It's the best I can do, I think.
Better data, better answer.
Yeah, right.
So Ryan has an early flight this afternoon, so we need to keep him on schedule. So let's thank him again for outstanding.
Thanks for having me.